Hello, everyone, and welcome to our symposium on convergent evolution and natural experiments. And we are really excited for this symposium to start, and we are happy to have really excellent talks ahead of us with a range of model systems and organisms that will be covered. And we are really proud as well that we will have not only one, but two speakers that have received an award during this conference. So congratulations again to Jerica Jameson and Scott Edwards, who, And Scott Edwards from the Harvard University in US will also be the first speaker of our symposium. And he will be talking about using phylogenies to identify genotype-phenotype correlations using convergent evolution.
Thank you very much, Hans. Sorry there's not enough seats in here, but there is couple open seats right here and a couple over there. So don't be bashful. Come on, relax. I thought, I'm very excited. Thank you, Hans and Ariana, for setting up this symposium. It's really wonderful to be here. And I thought I'd tell you about some of our recent work developing statistical models to link genomic and phenotypic variation using phylogenies. And it was actually at the Rhode Island evolution meetings just a few years ago that Stacy Smith and others and we were at a symposium on the use of phylogenies, and we were struck by the exciting but sort of heavy emphasis on ecology, biogeography. And we sort of got together and said, well, hey, we can use phylogenies to basically map genes for traits as well. And that's what we wrote a little opinion piece here. And there have been some very exciting examples of using phylogenies to try to find the genes underlying traits in recent years. The field probably has not coalesced yet, but there's a lot of opportunity for new contributions, new approaches. The basic goal of genetics, which is sort of illustrated on that column in the center, is to, you find a segregating population, you find markers, and you basically associate the markers and the segregating phenotypes. We can't do that for most of biodiversity, so we need to use other approaches. And those approaches are sort of outlined on the column on the right, where we can imagine finding a trait that is popping in and out of a phylogeny. Maybe there's convergent evolution. And we can imagine finding clades where the trait is moving, evolving and regressing. And that might give us opportunities to map genes for those traits. And then, of course, just as in classical genetics, we can follow up with functional experiments. And I'll illustrate an example of that at the end. So, in recent years, there's been different ways of looking at this problem. Some groups, like Gil Bejarano's group at Stanford, have been taking the approach of finding regions of the genome, non-coding regions especially, that have been deleted in lineages that have a certain trait or are missing a certain trait. And that's illustrated here. So you're looking for a physical deletion from the genome, whether in a gene or a non-coding region. Our group has taken a different approach where we're not looking for deletions, but we're more looking at accelerations of evolutionary rate, primarily of non-coding regions. Of course, accelerations of formerly conserved regions means that these regions are either losing their function, selections becoming relaxed, or they are achieving a new function, perhaps through adaptive evolution. And so those are the main sorts of approaches I'll be talking about today. And we have a growing number of software in the family called PhyloACC, standing for phyloacceleration. And this is largely work with Jun Liu's lab. He's a statistician at Harvard. And in the first sort of model, we can imagine a simple binary trait where we have lineages that we're particularly interested in. Maybe they're flightless lineages, maybe they have a certain plumage, or maybe they have a different number of appendages, who knows what. And we can sort of designate these as target lineages and then look for accelerations specifically on those lineages. We've expanded this to accommodate gene tree heterogeneity, since incomplete lineage sorting and things like that can induce false signals of acceleration when no acceleration exists if you force a wrong tree on a local gene tree. And then finally, I'll tell you a little bit about a model in phyloACC-C, which stands for continuous, where we're looking for associations between the rate of evolution of genes and evolution of a continuous trait. So in the basic model, we're trying to identify genes or non-coding regions that are accelerating in so-called target lineages, which you see indicated on the red branches. And the challenge here, as in convergent evolution generally, is to find genes that are accelerating just on those target lineages and not on the clade as a whole. Many software out there find it difficult to distinguish between clade-wide accelerations and accelerations specifically on target lineages that have arisen multiple times. So in the basic model, we have a transition matrix, Z, which allows branches to transition between three conservation states: the background state, the conserved state, and the accelerated state. And so you can imagine lineages that go from a conserved state to an accelerated state. In much of our work, we sort of imposed a Dalo assumption where once it's in an accelerated state, it can't revert back to a conserved state, but it's a very flexible model otherwise. And for each of these three conserved states, we're estimating rates of evolution either of the genomic element itself or of the phenotype. Now, the key, I think, to the model is that we employ a pair of Bayes factor tests which allow us to discriminate between that clade-wide acceleration and the lineage-specific acceleration. So in the basic model, in the gray box, that's a model of no acceleration at all. In M1, we have a acceleration specifically on one or more target lineages. And in M2, we have a free model in which acceleration can occur anywhere on the tree. And by applying these two pairs of Bayes factor tests, we can rule out, for example, clade-wide accelerations or accelerations that occur outside of the target group. And this, I think, is what gives phyloACC its sensitivity and precision. And so as a first example, we applied some data to echolocation in mammals. The echolocating lineages are indicated in the orange. And you can see on the two axes these two Bayes factor tests. So on the x-axis, we have Bayes factor 1, which is comparing the no acceleration model to the target lineage model. And you can see that points to the right. Each point there is a different conserved non-coding element, so it's a large number of points. We're interested in ones to the right of that line or whatever Bayes factor level you choose, as well as the y-axis Bayes factor 2, which are comparing the M1 model, the target lineage model, with the unrestricted model. And so the small cluster of points in the orange there are genes that fulfill both those Bayes factor tests, accelerating strictly on one or more target lineages. And so you can see that we can, with a modest number of initial seed inputs, we can get a large number of candidate regions that are accelerating specifically on the target lineages. So in the second test I mentioned, we call it phyloACC-GT for gene tree. This is accommodating gene tree heterogeneity. Most tests of rate evolution in phylogenetics ignore gene tree heterogeneity. And the problem is that if you force a wrong topology on a gene tree, that will induce a signal of false acceleration, a false positive. So incorporating incomplete lineage sorting is important here. And we did that through a kind of a creative proposal scheme in the MCMC of the test. And what we did is we sort of impute sequences down the local gene tree, which are indicated by the points in those trees. And we allow the likelihood of a given ancestral sequence to guide the proposal of the next gene tree. It's a really innovative approach developed again by Jun Liu and our collaborators, and it provides an efficient way to find the local gene tree as well as to estimate the rates. And what you can see here for 3 different acceleration scenarios where we have just a single acceleration or multiple accelerations, you can see in the orange that in the blue, that we get a modest improvement in performance, whether it's minimizing false positives or and basically the precision of the test. So a good 10%, we had a good 10% or so improvement in terms of ruling out false signals when we take gene tree heterogeneity into account. And this approach hasn't yet been widely adopted, but we did see some folks studying heliconius butterflies. You can see their alignments of non-coding elements on the left, and using this to look for lineage-specific accelerations on the branch leading to heliconius, which of course is a well-known adaptive radiation. I should say too that we've greatly improved the user friendliness of the whole set of software, and so it's a lot easier to use than it was a year ago. So in the continuous model, what we want to do now is correlate increases in evolutionary rate of the genome with shifts in a Brownian phenotype. And this is work by Patrick Gemmell. You can see we've looked at a data set from Nathan Clark's lab looking at longevity in mammals. You can see on the right lineages like humans and bats have greater than expected longevity for their body size. And you can see the distribution of maximum longevities in the red bars there. And so in this approach, what we're doing is we're assigning a sort of a phenotypic variance variable, beta 2 or beta 3, depending on the conservation state of the genomic element. And so the test is one where we're looking for looking at the ratio of beta 2 and beta 3. Those are scaling elements for the phenotype. And we're asking essentially whether that ratio or whether the ratio of the log of those two betas is different from 0. Because that's saying that there's some suggestion of an association between the rate of the branch and the rate of evolution of the phenotype. Importantly, it's not necessarily a correlation between, say, faster rates of the genome and faster and larger values of the phenotypic trait. The test can find associations between increases in evolutionary rate of the genome or decreases in the phenotype. It's really an association between the rates and not necessarily the directions of change. Here's an example of a conserved non-coding element on the left and the tree of that element, the normalized phenotypes in the center, and then the alignment of the element on the right. And the red branches here are branches that are accelerated for the element. And you can see, for example, on the top, those are things like shrews and stuff, which have a fast evolution of this element, but which actually have relatively short lifespans. But again, it's not an association of the directions of change. It's an association with the speed of change. And so that branch with the shrews and other rodents at the top is contributing to this success of this association. But so are the marine mammals at the bottom, which you can see. You've got some whales at the bottom. They've got really long lifespans, but and also faster rate of this genomic element. So both of those scenarios are contributing to the association between the genomic element and the phenotype. And so you can see, for example, on the left you've got the genomic rate of evolution of the genomic element with the red and showing the prior and posterior distributions. And on the right, you've got this ratio of beta 2, beta 3, the two phenotypic scalars, with the prior in dotted line and the posterior. And so you can see that posterior is shifted significantly to the right, indicating an association between the two. Okay, I'll end with a couple examples. The first is by Subir Shakya, who's a postdoc in the lab. He's taken advantage of the AVANET database to look for genomic associations with tarsus length. It's a very easy trait to measure in birds, and we have the trait for all 10,800 species. And you can see here the branches colored in red for lineages with shorter traits. Does that say short? Or in blue, which are, longer traits in red, shorter traits in blue. And so what we did is we ran Bayou, a well-known software package for looking at continuous traits, to find regions of the tree that were shifting dramatically in the rate of evolution of tarsus length. Here are 4 examples: penguins, bulbuls, swallows, and kingfishers. We missed hummingbirds, of course, because of the way we had to split the data up. This was hard to analyze the entire tree at once. But we basically focused on shortening of tarsus because it occurred more times in our analysis than did lengthening. So we were able to harness, and in most of our studies, we've looked at these regions called conserved non-exonic elements. These are basically regions of the genome that are highly conserved and yet are not in a coding region. These are likely to be regulators of gene expression. And so you can see this black region in the middle. It's showing high conservation despite not being in a coding region, and we can look for changes in rate of regions like this. And so Subir did the 2 pairs of Bayes factor tests and found a set of regions fulfilling both criteria, about 340 regions out of a total of about 14,000. These are regions that are changing in tandem. In this case, we actually are using the discrete, the binary test, sort of long tarsus versus short tarsus is the way we categorize the data. And what's cool about, well, a couple things. First of all, we're finding that a lot of the elements that are associated with tarsus length are lineage specific. They're not shared between different members of these 4 lineages of birds, but they do seem to be involved with the same genetic networks. So if we look at the genetic network for tarsus evolution, which is actually quite well known in vertebrates, we find that different genes in this network are sort of being flagged by different non-coding regions that are very close to them. And so you can see here that we've sort of indicated which of the 4 avian lineages are showing acceleration for a gene that is close to, for an element that is close to a given gene. And so you can see that there's a lot of sharing at the network level, although perhaps not as much sharing at the actual, at the level of the genes that are close to the elements that are undergoing acceleration. But we think that maybe the network level is the level at which most of this kind of convergence lasts and is manifested. So we've been doing a lot of generating new phenotypes. Although birds are really well studied, there's a lot of phenotypes for which we still don't have good information. And so natural history collections are being really important here. We're trying, and Subir is now using some of these approaches to look at the evolution of flightlessness in ducks, which are a really nice group to look at. So I'll just end with some of our work on flightless birds. And these, of course, are the quintessential non-model species. We can't do crosses with these things, but we want to know why they became flightless. And of course, flightless birds have a lot of phenotypes that are of relevance. They've got this, they've lost the keel on their sternum. They've achieved really big and sometimes very small body sizes. And of course, their forelimbs have undergone a dramatic reduction in many cases, or in the cases such as the moa, they've completely lost their forelimb elements. And so a few years ago, we sort of confirmed previous phylogenetic work showing that the tinamous, which of course are a member of this group but which can fly, were nested within the flightless radiation. This conclusion, which was found by other groups years earlier, really rocked the bird world. It was thought that there was a single loss of flight at the base of the flightless lineages, but it turns out that multiple convergent losses are probably a better scenario for loss of flight. And so we looked around the genome and we found about 280,000 of these conserved non-coding elements. And we applied, in this case, the binary trait test, the original phyloACC. We found a number of cases like this where you have a genomic element that is conserved throughout the history of birds and yet shows acceleration specifically in one or more flightless lineages, such as this example here showing acceleration in the MOA and in the RIAs. In total, a little less than 1% of our elements conform to this pattern. And we found that many of the genes that were close to these elements in the genome were genes that were already well known to be involved with developmental processes. So this was really nice confirmation. And it's also the case that we tend to find a lot more functional coherence of sets of genes that are flagged by the regulatory elements than by the genes themselves. In other words, if we look for acceleration at the gene level, we find a moderate number of genes, but those genes don't seem to have a lot of coherence in terms of their function. So this has led us to the conclusion that regulatory evolution is probably driving this particular phenotype. We conducted a TAC-seq to look at regions of the genome that were epigenetically exposed and active. And we have to do this because when we flag an element as accelerated, well, we don't know which aspect of this multifaceted phenotype is being influenced by a given element. When we can look at where that element is in open chromatin in the developing, in this case, chick embryo, we can immediately surmise what part of the phenotype, whether it's the keel, the forelimb, or other parts, might be related to this particular element. And so this shows that most of the elements we're finding are in regions of open chromatin. They're overrepresented in regions of open chromatin, which increases the likelihood that they are genuine enhancers. Now, we've been collaborating with Emma Farley of UC San Diego, who is an expert in so-called massively parallel enhancer screens. And what these are basically cocktails of several hundred enhancers, which we've been injecting into chicken forelimbs, and we can then harvest the mRNA that is produced by those constructs and basically figure out which of the several hundred enhancers we've injected is eliciting a gene expression response in the developing forelimb or hind limb. So we put these barcodes on there. And this is just what the data looks like. The input are the blue DNA constructs. You can see we've done replicates in multiple different parts of the developing embryo. And the red are the mRNA, each point being a different enhancer-associated mRNA. So some enhancers are driving gene expression, other enhancers are not, and we want to figure out which enhancers are. And for example, we found examples of enhancers that are able to drive gene expression in both the forelimb and the hind limb. That's what you see there in panel A. In panel B, we have examples that are just in the hind limb, and in the forelimb. And panel C is an example that is driving gene expression just in the hind limb. So it's really amazing that we can actually get tissue-specific enhancers in this classical non-model species. And this, I think, is a really nice way forward in terms of finding regulatory elements associated with different phenotypes. So some conclusions here. We've shown you a bunch of different ways of looking at statistical models for looking at non-coding evolution and associations with phenotypic traits. We've found a lot of success looking at non-coding elements, less success looking at proteins and genes themselves. And we have found that epigenetic tools like a TAC-seq are very useful for helping us narrow down the large number of enhancers to those that might be specifically involved in a phenotype of interest. So this has been a great collaboration between developmental biologists, paleontologists, bioinformaticians, and I'm sort of the lucky bird watcher in the back who gets to orchestrate things. But thank you very much. Questions?
Question. Whom that's the last one?
The question was, there are pros and cons to using a Bayesian versus a maximum likelihood approach. I mean, I think one possible advantage might be the issue of multiple testing. In other words, the requirement for corrections with a maximum likelihood test is probably less important with a Bayesian approach. But I think it's probably, Jin Liu is just a die-hard Bayesian, and so. Some of the gene tree model is quite computationally expensive, but it's basically done on an element by element basis, and so if you can parallelize things, it's not too bad. Yeah. Yeah.
So if a lot of the results you're getting are suggesting independent mechanisms, this is going to be an unpopular question in this symposium. I mean, convergence isn't really central to using these approaches, is it? Like you could do this on independent origin traits, and then you still have to do the functional work afterwards to connect the dots.
Yeah, you can do. I mean, I think in our case, for example, in the Tarsus case, we're not getting convergence at the specific non-coding element or even of the gene, but we are getting convergence at the level of the network. And so I think, and who knows, maybe genetic architecture is that way. We might expect that to be the null hypothesis. I don't know. But I mean, in terms of functional testing.
If you had some cool trait in one lineage, right? I mean, you could these accelerate, you could still do this kind of thing.
Yeah, absolutely. You can. Absolutely. If it doesn't have to be a convergent trait, but I mean, in lots of I think the power is going to be significantly higher. It'll be higher with a convergent trait. You'll miss a lot of associations if it's just a single acceleration. And timing is also important. You could have very rapid evolution, but it's so recent that you just don't see it even if something's been accelerated. So. Yeah, I think single origins and we just need to be. We don't know what the sensitivity of these tests is yet. So, yeah. Hey.
So most of the examples you gave were pretty deep in the trait, shortened tarsi in large lineages and whole families. Do you think this method would work better for those kind of situations where you have convergence in large clades, or would it work just as well or better in situations where you had a lot of quick evolution, within a clade towards the tips of the bird tree?
Yeah, that's a great question. We don't really know yet. We don't have a lot of examples. In the ratite case, those were all pretty deep, independent origins. Same with the Tarsus case, I would say. And I think we just need to see what's going on. I mean, one thing we don't know yet is whether a lot of the accelerations are the result of loss of, relaxation of selection, or whether they've just sort of, it's adaptive evolution to a new function. And for that, actually, population genetic information can be useful in discriminating that sort of thing. And so I think we just need to get out there and start looking at examples and push the boundaries and see where the sweet spot is for these sorts of tests.
Okay, our next speaker in this session is Maddie James from the University of Queensland in Australia, who will be talking about magic populations of Australian wildflowers and the role of chromosomal inversions on the genetics of convergent ecotypes.
I'll wait until 10, or I can just start. Perfect, wonderful. Hi, everybody. Okay, perfect. Yeah, that's good. Hi, everyone. Sorry, I'm too short. Hi, I'm Maddie. I'm from the University of Queensland in Australia. And I'd first like to thank the organisers for inviting me to speak in this symposium. And today I'll be talking about the role of chromosomal inversions during parallel adaptation in a native Australian wildflower. Of course. Sorry, I'll speak louder. Perfect? Better? Wonderful. I'd like to begin by acknowledging the traditional owners on the land on which we meet, as well as the Yugur and Turrbal people from Meanjin, where this research has taken place. I'd like to thank all of the members in the lab who've been involved in this research over the years. You can see us there dressed as our study system. Some of those people are in the audience here. So as we all know, nature is extremely diverse, and a lot of us want to understand how this diversity has arisen, so how populations have diverged to create this. So let's consider that process of population divergence, and imagine an ancestral population has given rise to two separate populations adapting to contrasting environments. So they are under divergent natural selection. So in grey, we can see the genomes. It might be considered easy for these populations to diverge if they do not exchange genetic material. This is because each population accumulates locally adaptive alleles, which enable them to adapt to each environment, and divergence occurs because of this adaptation or via drift. However, if these populations are exchanging genes, for instance, asymmetrically into one population, locally adaptive alleles are transferred and recombined into the recipient population, which makes them similar. So during such cases, we have this divergence of, during such cases of divergence in the face of gene flow, we have this tug of war between the homogenizing effects of recombination and the diversifying effects of divergent natural selection. So how do populations actually diverge when they're still exchanging genes? So in this scenario, divergence is facilitated if locally adaptive alleles reside in regions of reduced recombination. And this effectively traps these alleles together in a block, which prevents their breakdown and integration with alleles from the other population. And with this, natural selection can efficiently increase the frequency of the adaptive alleles that are in this block, as well as efficiently remove any blocks of maladaptive alleles. So there are two main mechanisms that can suppress recombination. We have modifier alleles, so genes that influence rates of recombination at specific locations in the genome, and this might manifest as these overall haploblocks of low recombination. And we also have chromosomal inversions, which are structural rearrangements of the genome where a segment of it is physically reversed. And with this, recombination is suppressed in inversion heterozygotes. There are a few other mechanisms, but these are just kind of the main ones of the focus of the talk for today. So back to our original scenario, these regions of reduced recombination that surround locally adaptive alleles might manifest as general haploblocks of low recombination or chromosomal inversions that differ between the populations. However, we're still kind of really largely unaware of the specific dynamics of how recombination can facilitate divergence in the face of gene flow. So we do not know how repeatable and predictable the evolution of inversions and haploblocks are, and whether these regions directly underlie adaptive traits. And this is exactly what we are uncovering in the system that we work on. So let me tell you about the system. We work on the Senecio laudus species complex that's distributed across Australia and contains multiple ecotypes. Two of these ecotypes, the dune and headland, they occur along the coast and often occur adjacently or in parapatry. So we have multiple of these parapatric dune-headland population pairs, as you can see on the map there. These two ecotypes have morphologically based genetic differences. So dune populations are tall, have large leaves, are found in the sand dunes, whereas headlands are short, small succulent leaves, and grow in the rocky headlands. And one of the main selective agents that differs between the environments is the levels of salts in the soil. So the headland environment has a lot of salt spray, and the dense soils retain salts, whereas the porous soils of the dune environment do not. In the system, we've also undertaken reciprocal transplant experiments, which has enabled us to uncover a key adaptive trait in the system, which is gravitropism. So I won't go into too many details, but gravitropism is the ability of a plant to bend or respond to that downward pull of gravity. And we've shown that it is adaptive and also associated with the phenotypes in the system. So tall dune plants do respond to gravity, and short headland plants do not. Another key aspect of the system is that these populations have arisen multiple independent times along the coast. So it's a system of parallel evolution. So our dune-headland parapatric pairs are often sister taxa in the phylogeny, and we have really strong evidence to show that each pair can be considered as a replicate of the evolutionary process. And finally, we've measured gene flow, and although contemporary rates of gene flow between the dune and headland of each pair is quite low, it is likely that early ecotypic divergence occurred in the presence of gene flow. So altogether, this makes Senecio ideal to help us answer our questions from before. So let me start with the first question. And to look at how repeatable and predictable inversions are, we identified them between the dune and headland populations of each of our 6 replicate pairs. I won't go into too many details on the methods, but we used a combination of local PCA, patterns of heterozygosity, and high C data to measure them. And we found quite a few inversions, 104 across our 6 replicate pairs. So in the x-axis are our chromosomes, and then we have our 6 replicates. Inversions are in pink, and we have a few haploblocks in green. So we see a lot of variation in the number and position of inversions across our replicates. However, we do see some regions of the genome that seem particularly prone to evolving these low rates of recombination, where we see a few regions where there's multiple kind of haploblocks and inversions there across our pairs. So this diversity of inversions might imply that there is no parallelism. However, one possibility is that even though the inversions capture different sets of genes in each of our replicate populations, maybe these genes share similar functions. So we set out to test for this parallelism at the level of the function. So to do this, we undertook a gene ontology enrichment analysis, where for each replicate pair, we took those inversions and haploblocks, got the genes inside them, identified which gene ontologies were enriched for each pair, and then asked whether there were any gene ontologies repeatedly enriched across all of our 6 replicates. And there were, so we found that the different inversions of the replicates capture functionally equivalent genes. So we have parallelism of the functions of the genes that reside within the inversions, which is super cool. So let's look at what those functions are. So we found 3 gene ontology categories that were repeatedly enriched across all of our replicates, and they all have to do with symporters, which are membrane transport proteins that transport ions across membranes. And what is really interesting is that symporters play a crucial role in both salt tolerance and gravitropism. So as we know before, salt differs between the environments, and when a plant is in a salty environment, it's important for it to maintain ion homeostasis to avoid salt toxicity, and this is effectively done by symporters transporting those ions and salts out of the plant. And as we know before, gravitropism is also a key adaptive trait that we identified. And again, for a plant to elicit a gravitropic response, it's all about how oxygen is actively redistributed in the cells, and again, that's all have to do with symporters. So here we're uncovering how these kind of molecular mechanisms not only relate to a key selective agent in the system, but a key adaptive trait which is associated with the overall phenotypic differences between the ecotypes, which is super cool. So if we remember, it's the inversions that are capturing these symporter genes. So here they are again. So if these inversions and haploblocks are truly involved in adaptation, we expect them to have elevated levels of differentiation between the ecotypes. So we looked for this. So I'll step you through this graph. At 0, we have our genome-wide DXY, which is our between population differentiation, and each bar is an inversion or a haploblock. Anything above the line has higher DXY compared to the average. Anything below is lower. So what we expect is that an inversion, or is that if an inversion is involved in adaptation, it's going to have higher DXY. And this is an example for just 2 of our replicate pairs. So for pair 2, we see a few inversions that are likely involved in adaptation, but for pair 4, we see many more. And now what is really interesting is that pair 4 has higher rates of gene flow. So this is everything we're kind of expecting from our theory that we were talking about before. So following on from this, we asked if higher rates of gene flow lead to a higher proportion of inversions capturing these locally adaptive alleles. And that is exactly what we found. So here we see that higher rates of gene flow between the ecotypes of a pair lead to a greater proportion of inversions having high DXY. And so this shows that as there is more gene flow in that divergence process, regions of low recombination become more important. So this supports all of the theory that we were speaking about before regarding when you've got more gene flow, there's more maladaptive alleles being integrated into the population. So then the role of inversions capturing these alleles of small effect is really important to make selection more efficient, which is, yeah, I think quite an amazing result. Okay, so we've just got one kind of final piece of the puzzle here is, do these inversions directly underlie adaptive traits? So what we've seen so far are associations between traits and these inversions, but we can make a direct link by using a genetic resource we've generated in the lab, which is a magic population. So I have a few minutes left and I'll tell you what it is. So a magic population is a multi-parent advanced generation intercross population. And to construct it, we crossed multiple individuals together from one replicate pair for 11 generations to create the magic population. And what this does is that these many generations of crossing and recombination breaks down both genetic and trait correlations. So this means we can isolate the genetic basis of any trait we want to an extremely high resolution. And we did this by undertaking a genome-wide association study on the magic population to uncover the genetic basis of gravitropism. So here is our GWAS, and what I've highlighted is just the GWAS hits, so regions of the genome that are strongly and directly associated with gravitropism. So we see adaptation here is polygenic. So the question is, do these hits correspond to those chromosomal inversions and haploblocks that were detected in this replicate pair that constructed this magic population? And the answer is, well, no, they don't, unfortunately. We only kind of see one haploblock or chromosomal inversion down the bottom capturing GWAS hits. But maybe this result is actually not so surprising. So the pair that constructed this population doesn't have high rates of gene flow, so maybe we're not expecting that association here. And we've only measured here one adaptive trait. Maybe we only see that association between the kind of GWAS hits and the inversions when you've kind of got multiple traits. So maybe inversions are only capturing locally adaptive alleles that are involved in kind of, multiple trait adaptation. And we'll soon be able to figure that out because we're doing a GWAS for another 30 traits in the system, so we'll be able to get this multi-trait responses to selection across all of our traits and see how that looks in terms of, recombination and inversions. Okay, so with that, our work in Senecio is enabling us to understand the dynamics of adaptation in nature. We're examining how genome features help facilitate adaptation, how natural replicates are helping whether there are multiple functionally equivalent solutions to adaptation, and our replicates are also helping us look at how repeatable and predictable evolution is, and whether we can kind of forecast adaptive trajectories into the future. And finally, our magic population is helping us make direct links between the genotype, phenotype, and fitness in a natural system. So again, I'd like to thank everyone in the lab who's involved.
In your replicate population 6, it looks like there was only one or two inversions. Is there something different going on in that particular transect?
There's a few things with replicate pair 6. The sample size is a bit lower than the others, and also that pair is the least divergent as well. So we might expect something there as well. So there's not as much contrast between the ecotypes.
Thank you.
Definitely.
Hi. Hey, sorry, here.
Hi.
I was curious, maybe I misunderstood something, but the gene ontology analysis you did it allow you to identify if there were certain functions co-occurring within the haplotypes, as if they were sort of protecting them from being
We haven't looked at that.
Disrupted.
We haven't looked at that yet. This
okay.
is all kind of really new analysis, but it's something definitely I think we should look at. But we haven't looked at the kind of between haplotype functions and how they kind of link together with each other.
Yeah.
But super interesting.
Yeah,
Definitely.
thank you.
Thank you. It's about time. I'm going to probably leave, I think.
Okay.
Oh, feel free to ask more questions, but I think I would have been on time there.
Okay, as the last speaker of the session, we have Zoe Hert from Indiana University in the US, and she will be presenting on the genetics of convergent evolution in orange tomatoes from the famous Galapagos Islands.
Hello, I'd first like to thank the symposium organizers for inviting me. I'm really excited to be part of this symposium. And just to start off with some broader context to this system, so there's exceptional diversity in fruit color across Solanaceae, but especially in the genus Solanum, which includes nightshades like tomato, potato, and eggplant. And this diversity is not just qualitative, but in fact there are over 100 transitions in fruit color across the genus. Despite this fact, fruit color has received relatively little attention from evolutionary biologists, much less evolutionary geneticists, compared to traits like flower color. And so, in order to begin understanding both the evolutionary and genetic mechanisms shaping fruit color, I want to look at one case of repeated fruit color transition, which is the 3 wild tomato species that live on the Galapagos Islands. So these are Solanum galapagense, Cheese maniae, and Pimpinella folium. And so all 3 of these species are quite recently diverged, and Galapagense and Cheese maniae have this like derived orange coloration, whereas Pimpinella folium has an ancestral red coloration. And Galapagense and Cheese maniae are both endemic to the Galapagos Islands. As you can see in these images, they have these like really bright orange colors that sort of range from like a yellowy orange to a deeper orange. And on the mainland, Pimpinella folium is distributed from Ecuador through Peru down through Chile, and it has this like really vibrant orange color. However, very recently, Pimpinella folium has invaded the Galapagos Islands, and exclusively on the islands, Pimpinella folium has this convergent orange fruit color. And so both of these images are taken from the same population. And so because we only see these orange fruits on the islands, there are 2 competing hypotheses for why this trait emerged, essentially. The first being that the islands have relaxed selection for red fruit color relative to the mainland, so these orange colors evolved via drift. And the second is that this orange fruit color is due to strong positive selection exerted by the novel conditions on the Galapagos Islands. And so in order to test these hypotheses, as well as to answer a range of questions about this system, my lab has developed or has generated over 200 low-coverage genomes across 4 types of populations. So one is putatively pure Galapagense populations. Then another is Galapagense, Cheese maniae, and putative hybrids. We have also putatively pure Pimpinella folium populations, and finally populations containing Pimpinella folium, Cheese maniae, and putative hybrids. And so with these genomic data sampled at the population level, we can address a number of questions about these species. For example, we can understand genome-wide patterns of diversity, and we can also perform a number of population genomic analyses to look at genetic structure. And so these aren't the focus of my talk, but I just wanted to note some key context about this system, is that 1, all species harbor low genetic diversity, regardless of whether they are endemic or invasive. And of course, there's slight elevated diversity in hybrid populations. And second is that they're structured both by geography, based on what island they're located on, as well as species identity. And so today I'm going to describe how we are using these data to better understand the evolutionary genetic mechanisms of this phenotypic convergence. And I'm going to start with the genomic history of selection on carotenoid pigmentation loci. And so what's really powerful about this system is that we actually really well know the genes that underlie this like red, orange, and yellow fruit coloration, as well as just red, orange, yellow colors in general. And so pictured here is the carotenoid biosynthesis pathway. In bold are those genes in the pathway, and then I have pigments in these colored blocks. And the color of those blocks corresponds to the color of the pigment. So for example, lycopene is a red pigment, and it is pictured in red here. Previous work in our lab has looked at ancestry tracks in 2 Pimpinella folium populations, and those results are pictured here. And the main point of this result is that for one Pimpinella folium population shown at the top, orange fruit color is shaped by introgressed Cheese maniae alleles at this Psi1 gene. And in a second population, this orange fruit color is shaped by introgression of Cheese maniae alleles at a Psych-B gene. And so these genes are going to be a major focus of my talk. And so with this new data our lab has generated, I've looked at patterns of nucleotide diversity across these genes. And so along the x-axis of each of these plots is genomic position. Across the y-axis is pi estimated in 1 kb windows. And then also in the plots, each of the colored lines represents estimates for a given population. Galapagense populations are colored in green. Pimpinella folium are colored in blue. And then gray lines represent the location of the coding region. And those black lines going horizontally across the plots represent the interquartile range of genome-wide nucleotide diversity. And what is important here is that essentially in Galapagense, so looking at those plots on the left, nucleotide diversity is low relative to the genome-wide median, especially in Psych-B, which is suggestive of historical selective sweeps. And then in Pimpinella folium, comparatively, Nucleotide diversity, like statistically, is also low relative to genome-wide averages, but there are a handful of populations where there is strongly elevated nucleotide diversity. And that elevation is also like, the nucleotide diversity is elevated in Pimpinellifolium also relative to Galapagensii. And so this result isn't particularly surprising. As I've described previously, Pimpinellifolium is polymorphic for fruit color, so this genetic variation for fruit color is still segregating in the populations. With these data, we are not just restricted to these 2 genes, however. We can also look at a number of genes across the pathway. And so here I'm showing essentially the same plots, except I've also included ZISO and ZEP, which are 2 other genes that are not yet known to be like involved specifically in orange coloration, but are involved in the pathway. And both of these genes, like Cy1 and Psych-B, have low diversity relative to genome-wide patterns, but there is no statistically significant difference in their diversity between Galapagensii and Pimpinellifolium. So this result suggests that ZISO and ZEP are not involved in this novel orange fruit coloration, unlike Cy1 and Psych-B, which both are different between Galapagensii and Pimpinellifolium and have fruit-specific functions. And so in the next part of my talk, I wanted to look at specific coding region variants in Cy1 and Psych-B and connect those patterns of variation directly to the phenotypes. And so I extracted coding region sequences from Cy1 and Psych-B for every single individual we sequenced, and then I plotted that variation for both genes on one PCA, and then I colored each of these points according to the individual's corresponding fruit color. And so what we see is that there's no clear clustering by fruit coloration, and so this isn't wholly surprising because we already know from the introgression work I previously described that we can have independent changes in Cy1 and Psych-B that alter fruit color. In other words, a given fruit color is not governed by having a particular Cy1 genotype with a second Cy2 genotype. They can independently influence the trait. And so next, I wanted to look at each gene's coding variation independently. I'm going to focus on results for Cy1, but for Psych-B, we found that there was some partitioning by fruit coloration, but it seems to mostly be driven by cis regulatory variation, which I can talk about more later on if you'd like. But going ahead with Cy1, what we see is that orange and yellow colors cluster along that second principal component, and this second principal component is explained by 2 SNPs in Cy1. Both of these SNPs are non-synonymous, and on the right, I show their position in that Cy1 like genomic region alongside estimated pi across the region. And so what we can see is that for Galapagensii, nearby these 2 SNPs, there's very low nucleotide diversity. Cy1 in Pimpinellifolium, genetic diversity is also low, but there are a few populations where it's elevated relative to other populations. Drawing your attention to the graph on the left, here along the x-axis, what we see are the different genotypes for the 1081 SNP. Each of the colored lines on the plot represents the genotype at the 86 SNP. Along the y-axis is the individual's fruit color, and then each of those little points there are different individuals colored by their species/population. And so what I just want to highlight here is that orange fruits, which are clustered in this like bottom quadrant, are predominantly governed by having a homozygous G genotype in 1081 and heterozygote or G homozygote in the 86 SNP. And so in our data set, these 2 particular genotypes together are never associated with red fruits. And therefore, it seems possible that these 2 non-synonymous variants are causal. However, some orange-fruited individuals have different genotypes at these 2 loci, which could be explained by the contribution of another gene. So we essentially have orange-fruited individuals that the orange fruit is caused by variation in Cy1, but again, there's other genes that are likely contributing differently in other populations. And so taken together, these results in concert with the analyses of selective sweeps, as well as previous work from our lab on introgression, suggest that multiple genetic mechanisms may explain convergent fruit coloration. I think a lot of this story is still yet to be discovered, as we know that this phenotype is clearly complex among pimpinellifolium populations, so it's also probably complex among Galapagos populations. We just haven't had the chance to look at those genes yet. And so thinking about how multiple genes in this pathway may be contributing, I also just wanted to look at what factors may be shaping molecular variation across the carotenoid biosynthesis pathway. And so there's a lot of classical work already done on these sorts of questions, especially in the anthocyanin biosynthesis pathway. And it's been found that molecular variation can be constrained both by pathway position, so upstream genes are more constrained than downstream genes, as well as by functional class. So structural genes experience far more selective constraint than regulatory genes. And so I've essentially repeated some of these analyses using carotenoid genes. And so first, I looked at relative pathway position of all of my genes in this pathway, and then calculated non-synonymous to synonymous polymorphism ratios for each. And what we see is that regardless of whether you're looking at Galapagos data or pimpinellifolium data, there seems to be no clear relationship between relative pathway position and selective constraint. And this may be expected because the carotenoid biosynthesis pathway is nonlinear. Unlike anthocyanins, you have the production of these pigments in relatively intermediate positions of the pathway, not just at the end. Next, I looked at variation between structural genes and regulatory genes. So most of these regulatory genes are just a handful of transcription factors that have been identified as being associated with carotenoid production. And this, again, is non-synonymous polymorphism and synonymous polymorphism. And what we see is there's essentially no difference in selective constraint between regulatory and structural genes affiliated with carotenoid biosynthesis. There is one last factor that can influence selective constraint that I looked at, which is gene duplication. And so gene duplication can relax selective constraint on one or both copies of the duplicated genes. And the carotenoid pathway is a really good system for addressing this question, as there's multiple different gene duplications throughout the pathway. And so I essentially looked at polymorphism and divergence for the genes in the pathway using the highlighted genes as duplicated genes, and then other genes in the pathway as non-duplicated genes. And what we find is that for non-synonymous to synonymous polymorphism, there is not statistically significant, but still elevated non-synonymous variation in pimpinellifolium duplicated genes. And then looking at divergence, I compared Galapagos and pimpinellifolium each to another wild tomato species, Solanum pinellii, and again found relatively elevated ratios of divergence in duplicated genes. And so this elevated non-synonymous variation seems to be evidence of relaxed constraint in duplicated genes. Furthermore, these gene duplications may have allowed for some tissue-specific specialization that we already know of. So for instance, Cy1 has a function specialized to fruit tissues, whereas Cy2 is specialized to leaf tissues. And so all in all, in each part of my talk, I addressed one part of these questions. I'm just going to go through this quickly because I know I'm short on time. But essentially, in the first part of my talk, I showed how past selective sweeps are evident in Galapagos, whereas variation in these pathway genes is still segregating in the polymorphic pimpinellifolium. Multiple different genetic mechanisms seems to contribute to orange fruit color convergence. And finally, when we look at selective constraint in this carotenoid biosynthesis pathway, gene duplication seems to be a major player. And so finally, I hope to expand these sorts of analyses across the genus Solanum to better understand that diversity of fruit color I described at the start of my talk. I think it's really important to both look at this more micro level analyses as well as the macro level analyses across the group. And just finally, I'd just like to acknowledge my lab, as well as our collaborators and our funding resources, and again, the symposium organizers. I'm happy to take any questions.
I have a question. Do you know if there is any selective consequence of being more on the red side,
So
of being a tomato?
yeah, so a lot of our work there is still quite preliminary, so I don't feel comfortable giving like a strong like yes or no to that. What I think is happening is that birds on the islands probably have different preferences for different fruit colors relative to the mainland.
So where does the cheese manii, I'm sure I'm saying that wrong.
No, you're okay.
And also the hybrids fit into this role? Because I know that it looked like, if I recall correctly, they had different hues of orange as well.
Yeah, so I haven't had a chance to quite analyze that data yet, but I plan to look at these like PCA type analyses, looking at more so the full like range of variation of color as opposed to just like binning things into slightly broader categories. And our cheese mania sampling isn't as strong, so a lot of these populations are more isolated, and the ones that do come into reproductive contact with like Galapagos and Pimpinella folium are like sort of hybridized out almost.
Thank you.
Okay, then
Thank
thanks
you.
again.
Okay, we will start. Thank you for welcome back or welcome. So our next speaker is Zoe from the University of Queensland in Australia, and she's going to talk about gene networks and gene expression and Australian wildflowers.
Okay, so hello everyone. Thank you so much for coming to my talk today, and thank you to Ariadna and Hans for inviting me to speak in this symposium. My name's Zoe, and I'm a PhD student from the University of Queensland in Australia. And if you saw Maddie's talk this morning, yes, we are in the same group. So today I'll be talking to you about some of my PhD work so far, looking at genetic networks and how they're associated with adaptation in an Australian wildflower. Before I begin my talk, I'd like to briefly acknowledge the traditional owners of the land on which this work was conducted. In Brisbane, this is the Yuggera and Turrbal people, and I'd like to pay my respects to their ancestors and descendants. I'd also like to acknowledge the Indigenous nations of Montreal. So, to understand how adaptations evolve as populations occupy different and novel environments, we need to understand how interactions between genes occur at different levels of organisation. For decades, we've investigated simple traits, where we look at the connection from DNA to a phenotype or a trait. But what happens when we provide a more holistic view of complex traits? Well, we find that networks occur at each layer of organisation, such as gene-to-gene interactions, but that these are not enough to completely explain trait variation. The genotype to phenotype map includes intermediate layers like gene expression, which is important to study as it associates genetic variation with phenotypic variation. So the expression of genes can be affected by space and time, and different amounts of expression are expected to be found throughout development and based on environmental conditions. As the expression of one gene changes, it can affect many more downstream genes, and when we put all this information together, we can create networks that look like this of interacting genes. These networks are often redundant, where different allelic combinations or completely different sets of genes can produce equivalent phenotypes. And one major aspect of these networks to consider is pleiotropy, which we often think about as one gene affecting multiple traits. But pleiotropy can also arise at the network level, where the same interacting genes can underlie multiple traits, and traits can manifest across multiple stages of development. So in this case, we can think of multiple interacting genes affecting multiple traits over time. We refer to this as developmental pleiotropy. Some examples of developmental pleiotropy have been found in diptera and pipefish, as I show here, where the same genes affect different aspects of development. And this is also important in plants, as they rely on the amount and distribution of hormones throughout development. There are hormones that affect embryonic traits, but through regulatory differences can also affect late-acting traits, such as flowering in mature plants. And these hormonal pathways, such as auxin, ethylene, and gibberellin, are often good candidates for affecting phenotypes through development. Some of these networks are simple, like this one depicting hormonal interactions underlying branching, whereas others can be very complex, like those controlling a suite of traits throughout development through complex genetic interactions. But most of these networks have been developed in model organisms, and it remains challenging to build these networks in natural populations. one powerful way to study genetic and regulatory networks in natural populations is to study the independent and repeated origin of an adapted trait. So, as populations diverge due to similar selective pressures, this can lead to replicates of evolution. Natural selection is deterministic, and by studying replicates of adaptation, we can study molecular evolution. Let me introduce a system where we have these replicates. So we're using Senecio laudis, an Australian wildflower that can be found on the coast of Australia. And Senecio can be used to investigate the molecular mechanisms underlying evolution, as it has independently adapted to the same environmental conditions multiple times. And we've shown local adaptation to these two environmental conditions, the dunes and the headlands. As you can see over there, as Senecio is adapted to the dune and headland environments, it's evolved two contrasting growth forms: a tall population that grows on the sand dunes and a shorter population that grows on the windy headlands. Multiple traits contribute to these growth habits, but many of these traits are ones that are largely controlled by hormones. And these traits have been replicated in dune and headland populations many times, and we want to know if the origin of these ecotypes is driven by changes at the nucleotide, gene, pathway, or network level. And so previous scans across these populations of Senecio have looked for genetic divergence leading to these parallel phenotypic traits. And interestingly, most of the parallel divergence sits at the pathway level, not at the gene or nucleotide level. This is illustrated in this figure here, where the parallelism is maintained by the biological function, shown in red, but decays at the nucleotide and gene level, shown in blue and green. Interestingly, most of the genes that were related to parallelism in Senecio were related to hormone pathways. So we think that hormonal regulatory networks control adaptive divergence. To test this hypothesis, we're using MAGIC, or a multi-parent advanced generation intercross population. We used multiple families of dune and headland individuals from one of the natural populations and crossed them for 11 generations. The shuffling of the two populations' genomes allows us to isolate the specific genetic architecture of a trait, as causal genes underlying the trait of interest are randomly placed on a common hybrid genetic background. Previous selection experiments have revealed that a hormone-dependent trait, known as gravitropism, is divergent between dune and headland populations and responds to selection in nature. The headland individuals don't respond to the pull of gravity, and these plants are known as agravitropic, while the dune individuals are highly gravitropic and respond strongly to changes in the direction of gravity, such as when they're turned 90 degrees. This trait is known to be adaptive in Senecio from previous reciprocal transplant experiments. So tropisms like gravitropism are important developmental traits that allow plants to quickly respond to changes in their environment. They are the ability of a plant to move in response to external stimuli, and these are often controlled by plant hormones. Gravitropism is when the plants respond to changes in the direction of gravity through various mechanisms, such as cell elongation or expansion on the basal layer of the hypocotyl, which can be initiated by an auxin gradient, as you can see here. So gravitropism is one trait that auxin is involved in, but we also find auxin plays many roles in many traits throughout development. And we have genetic data linking auxin to gravitropism, so now we can use this trait to find out if more levels of organisation are important for adaptation and to understand the connections between the levels better. So to investigate the link between genotype and phenotype, we looked at gene expression divergence related to gravitropism. We extracted RNA from the most and least gravitropic families for sequencing across 5 time points, and this is to capture the whole gravitropic response. So we identified 467 differentially expressed genes between the gravitropic and agravitropic plants, but we also used these differentially expressed genes to look at their patterns of gene expression over time and do weighted gene co-expression network analysis. This analysis creates networks where closeness between genes indicates similar patterns of expression over time, and we were able to identify clusters of genes within the networks related to plant hormones. So auxin, as expected, is the largest group there, but we also found ethylene and gibberellin. And we also found signs of crosstalk between hormonal pathways. For example, here we can see the co-expression network from gravitropic plants, in which we were able to see a connection between auxin and ethylene genes, which was not found in the agravitropic plants. And now we know there's a link between DNA and phenotype mediated by hormonal divergence. However, co-expression networks only identify potential relationships based on expression. But what if we want to know the directionality to help us build these networks? So to help add directionality, we looked at expression QTLs. Like QTLs that you'd obtain from a genome-wide association study, eQTLs provide potential correlations between a genetic variant and a phenotype. However, here the phenotype is the gene expression. So we have a range of SNPs, and we want to see if the expression of any given gene is likely to be affected by that SNP. With this analysis, we found 167 eQTLs regulating the expression of 193 genes. This figure here shows the expression over time of the genes affected by an eQTL. On average, we can see that the expression within gravitropic individuals is higher over time, particularly at the start of the gravitropic response. So the agravitropic plants produce less expression while the gravitropic plants produce more. But the timing of this is also important, as the gravitropic treatment individuals show a rapid decline in expression later in the response. Another important aspect to consider is the location of these eQTLs. And we can visualize eQTLs by looking at the location of that eQTL against the location of its target gene. If an eQTL is acting in cis, or from nearby in the genome, like in this example, we would expect to see a direct correlation between the two. And that is what we found in chromosome 1 and many other chromosomes. But sometimes we can see clusters of genes, such as in chromosome 19 and highlighted in blue there. So this clustering can indicate that a single eQTL may be affecting many genes at once. And interestingly, the genes within one of these clusters all have the same function, as they're involved in lipid protein transfer. So we now want to know if we can use the replicated populations to study the evolution of this network. One aspect of this is if the genes in one population pair are divergent in other population pairs. So we want to find the genes that have consistently evolved differences across the pairs, as these are candidates for genes under natural selection. So we want to see if there's sequence divergence across the natural population to find parallelism at the network level. If the eQTLs are identified across multiple population pairs, it could indicate that the overall network is being used to create parallel phenotypic differences. So we looked at the 6 population pairs outlined in blue and measured sequence divergence as DXY. DXY is a measure of the differences between genotypes from different populations, and we expect to find elevated DXY in genes that are divergent across these population pairs. When we looked at DXY, we found 20 eQTL target genes with DXY elevated above the expected amount. Of these, only 2, the ones that are shown in purple over there, are consistently high across all population pairs. And we can look at the functions of these 2 genes to see what they're involved in. So these 2 genes were found to be related to protein transport throughout the plants, salt stress responses, and adaptation to harsh environments. These are important candidate genes for parallel divergence that connects the genotype and phenotype through regulatory patterns. So we're consistently finding potential candidate genes related to other traits, like cell elongation, salt tolerance, and protein transport, indicating that these genes may be pleiotropic and involved in other traits. When we looked for genes related to gravitropism, we often find genes involved in other auxin-related functions. These genes may be co-opted for gravitropic responses. So these eQTLs that are related to divergence in gravitropism are good candidates for understanding parallel phenotypic evolution. Just to finish up, we have found that hormonal pathways are highly pleiotropic, and the evolution of these networks can have wide-ranging effects. Regulatory evolution links the genotype to phenotype map by controlling hormone signaling, and developmental pleiotropy is an important concept to consider when learning about adaptation. And just to finish up, I'd like to thank my lab and my advisors, particularly Daniel, and this is still a work in progress, but I'm very happy to chat or take any questions.
Any questions? I have one question.
Oh, yep. I was going to go.
So for this one EQTL that you showed that was shared, do you know if it arose independently or whether it spread by gene flow? Do you have any idea?
That's a great question, and we'd probably want to look into that more. But we know that these populations have evolved these traits independently, so we would assume that with the independent evolution of these traits, it's possibly different adaptations in the genes that are arising.
Okay, thanks.
Thank you, everyone.
So our next speaker is Catherine Elmer from University of Glasgow. She is presenting instead of Hongxin, who unfortunately could not travel, and they are using single-cell transcriptomics to study evolution of viviparity in common lizards. So thank you.
Yeah, okay. Thanks very much for that introduction and for having me for this symposium. And yes, I'm sorry that Hongxin couldn't be here because of some visa delays, but I'll do my best to present this work that he's been doing and we've been doing together on the common lizard. So viviparity is a fantastic trait to study for the context of convergence because viviparity is live birth, so giving birth to fully developed young, as opposed to oviparity or egg laying, which is the ancestral form, when of course we have eggs that we're all familiar with. So in the mammals, we have had one origin of viviparity, the placental mammals, of course including ourselves. Birds have always stayed oviparous. But squamates are an incredible system because they have many, maybe more than 100 independent origins of viviparity that have happened in that group. And so we can make comparisons between closely related taxa without a lot of loss due to evolutionary distance. And so thinking of this complex trait of viviparity, of course, viviparity includes many different aspects that that have to change all in concert, all in a suite of traits. So here we imagine we have the oviduct that encloses the egg with the embryo inside, and here we have an egg with calcification. And so we need a reduction of this calcification layer. We need a reduction of the eggshell and a thinning of the membranes. And then this has the embryo then within this egg sac. And so other aspects that need to change then is incubation. So under viviparity, the embryo is retained for a longer period in the mother. And so there needs to be transport through these membranes, through in the case of squamates, through the oviduct that didn't happen under oviparity and that enable the embryo to develop. So nutrients, oxygen go in, for example, and waste products and ions need to go out. And in order to do this transport, we need it needs a development of vascularization, so more blood vessels to be able to handle this transport. So vascularization is another feature. And then there's a suppression of the maternal immune system to retain this embryo within the body. So these are known to be a suite of features that have to all happen together. So it's a very interesting genetic system for how this can step, and if it does so in a convergent manner across different lineages. But so far we're a bit far from this as far as convergence. There's a lot of many different systems to investigate, but we're just at very early stages of understanding these genetics. So the species I'm talking about today is called the common lizard, Pseutoca vivipara, and it has a recent transition to viviparity from oviparity, probably the youngest transition within a species. And some work that was done by Hans some years ago showed particularly the recentness of this transition and looked in detail at the oviparous and viviparous taxa. So its name is the common lizard, but I like to think of it as the uncommon lizard because it's the only one, or one of very few, I should say, species that has genetically fixed oviparous and viviparous lineages within a single species. And so it's distributed across Eurasia. We can see on this map, but most populations are viviparous, and then there are some oviparous populations in the southern portion of the range. And so obviously here we have an excellent opportunity to identify some of these features associated with this trait. And so again, some work from Hans from some years ago showed that we had an ancestral oviparous lineage in this species, then a derived viviparous clade, and then a further putatively derived oviparous lineage, a clade that is an isolated population in the south of the range, the southwest. So it's called the western oviparous lineage. So we have two distinct oviparous lineages in this group. And this is an example of the staging of embryos that are either from the ancestral oviparous population or the viviparous. Then they are fully developed little baby lizard neonates. And the derived or western oviparous population that lineage has a slightly thinner calcified eggshell compared to the ancestral oviparous, and is oviposited by the female at a slightly later stage of development. But whether this is due to some environmental variations in the natural populations that had been studied wasn't clear, or how this represents possibly a feature of this reversal that has been hypothesized back to oviparity. I'm not going to talk a great deal about that aspect today, but we are going to focus instead on some of the physiological and molecular mechanisms that are associated with this transition from oviparity to viviparity using this system. And then looking for whether there's evidence of different types of oviparity within the species. And if this could be suggestive then of this breaking Dollo's law, reverting back to a complex trait that's been lost, or if it's some other type of transitional form between oviparity and viviparity. And so to do this, we needed to, for the first time, take this Zutoko model that we've been working on and now try to bring it into a controlled comparative environment. So we did a common garden experiment using this ancestral oviparous lineage, the derived oviparous lineage, and a representative of the viviparous clade, the derived viviparous. And so the first step then was to collect lizards from their natural environment, and thanks to the hard work of many field collections, we had populations from each of these different types. And then we brought them to a CNRS station in Molise, where we have excellent facilities for rearing these lizards in a common garden situation, both with outdoor husbandry and indoor husbandry, where we can monitor them. And so to study the reproductive phases and the pregnancy, we use ultrasound, and ultrasound the females during their reproductive stages. And so you can see we can monitor then and identify when, for example, ovulation has occurred and the size of the eggs, which is, I mean, the resolution is quite amazing. The lizards are only about this big, and you can watch the embryos develop within. And so to do this, then we did sampling at two different time periods. Vitellogenesis, so before pregnancy, and then during pregnancy. And what I'll present today is work from histology, what we'll call bulk RNA-seq, or sort of regular RNA-seq, as well as single-cell RNA-seq. So first we're going to look at this phenotype, this oviduct morphology, which we know is critical for reflecting the transport and communication between the mother and the embryo. So during vitellogenesis, so before development of the embryos, the ancestral oviparous, the derived oviparous, and the viviparous are shown here with H&E staining. And the key points I want to identify for you are we have a much thicker gland layer. That's these large cells that we see in the oviparous, as well as both types of oviparous. And these are much reduced in the viviparous. And in contrast, the muscular layer in the viviparous is much thicker, and it is thinner in the oviparous. So we can see this sort of qualitatively from these images, and as well can quantify it and show that the oviparous has more gland cells, more glandular features, and less muscle relative to the viviparous, and that the ancestral oviparous and the derived oviparous, the two oviparous lineages are very similar relative to viviparous. And this is the same feature during pregnancy, but the same pattern. So then I went to look at this single cell data that we have. So to do single cell data, we took, again, from each of our three lineages and from both stages. And we identified a number of different clusters, gene clusters. So this is just an overview to show you. But we identified a set of cells that represent gland cells. And we identified a set of cells that represent vascular endothelial cells. And I know that these are very difficult small density plots, so I'm just going to show a couple of aspects in which we can show that these cell proportions differ when they're quantified between the two parity modes. So cell gland-related features, we can see these shown in red and with the arrows, and that these are the cell gland-related features are greater in the oviparous than the viviparous. And then vascularization cells, which we can show here in the green, are greater in the viviparous individuals than the oviparous, consistent with our expectations from the development of this trait. And then single cell can look at these data as far as the numbers of cells, but to actually look at the proportion of these within the tissue, then we did deconvolution of RNA-seq data to measure more precisely the differences within the cells. And we can see then that we have shell gland-like features are more abundant in the oviparous populations, both during vitellogenesis and pregnancy, and that in this case the derived is more intermediate at molecular level than was seen from the phenotype. And similarly, consistent with our overview from the single cell data, the vascular endothelial cells are much more abundant, high proportion in the viviparous oviduct. And then third, I'll show this aspects of looking for the functional molecular mechanisms, so burrowing a bit more in detail into these gene groups. So we did a gene co-expression analysis, which many people will be familiar with. So look at the networks or the sets of genes that are co-expressed using WGCNA. And this is based on the bulk RNA-seq, looking at modules that are associated with parity mode and lineage and pregnancy. And I'm just going to highlight a couple of examples from this that we burrowed into with a bit more detail. So here are the colors that represent, the colors are arbitrary, but we have some modules of particular interest that were positively associated with viviparity, and some modules that are positively associated with oviparity. And so when we look at these among individuals, in our different groups, we can see that, for example, blood vessel development is a highly significant cluster for viviparity, and this has, is highly elevated in during pregnancy and vitellogenesis in the viviparous. And it's associated with enrichment clusters that are related to the development of like angiogenesis and the development of blood vessels, for example, that we see on the other side. Then we also had our other major feature was that of gas and ion transport, which is also elevated, particularly we found during viviparous pregnancy. And in this case, we have GO enrichment for ion and homeostasis-related GO terms. I know there's a long list there, but many of them in detail show these features. And then in the other direction, again, the expression of immune system during pregnancy we see is elevated in the oviparous and suppressed in the viviparous. And I'm sorry, this one did not come through, but again, it had consistently showed this same GO enrichment related to immunity terms. In conclusion, we showed that this oviparous and viviparous oviduct differs in morphology, with more gland cells in the oviparous and more muscle in the viviparous, and that these differences are then also reflected in the cellular proportions that we can see within each of these tissues, with cell gland-like cells more abundant in oviparous and vascularization more in the viviparous. And I'd say we showed that in structure, we find that the two oviparous are very similar, but at the molecular level, there are greater differences than are seen at the structural level. And these are reflected in the cellular features of gene expression. So overall, we see in this viviparity, this gas and small molecule transport being elevated, vascularization elevated, and immune system genes suppressed, just as we expected from our suite of traits that work together. And so these are evidence for these important mechanisms from the transition from oviparity to viviparity. And so now we're working more in detail to identify the history of these genes and many different aspects of these evolutionary differences between the modes. So this is a research project that's still in progress. And so I'd like to thank everyone very much who has helped with this project and our funding sources. Do I have time for a question? Yes. So, yeah.
One for
Beautiful results, and I think this is super interesting. I'm wondering, what do you think are, at the genome level, are the changes mostly occurring on the regulatory regions or maybe in the protein coding genes, especially since you have like this derived oviparous phenotype, so that requires something to be a little more evolvable.
So we definitely see a lot of regulatory differences and a lot of gene expression differences, and what we're doing right now is what you're suggesting. So we're working at the genome level to look at this moment. So we have crossing experiment, and we have the sequence analysis for both of those. So I'll be able to present more on those soon. Okay, thanks.
Thank you. So yeah, this talk is about invasive mosquitoes, and if you've ever wondered how it is species of mosquitoes and other taxa are able to get all over the world, well, this figure shows 1 year of global shipping. So you can see that pretty much every single coastal area in the world is just plugged into this enormous network that is a continuously functioning network. Things are hitching a ride on the network, and this process is serving as it's like a continuous redistribution of global biodiversity. And one species that's done really well from that is Aedes aegypti. This is, from a human perspective, a catastrophically bad species. This is the main vector of dengue, and it's also a very successful invasive species. So what we know from the last 500 years of its invasion history is something like this. It starts off in Africa. It invaded the Americas during the transatlantic slave trade. In the early 1800s, it got into the Mediterranean region. And then in 1869, the Suez Canal opened up, and from there it invaded the Indo-Pacific region. Now, if you look at a map of individual genetic variation, that lines up pretty nicely with these invasion histories. We see a lot more variation in the native range and then less everywhere else. Why I'm showing you this introduction is I want to really get across this one point, which is that by the end of the 19th century, the invasion is pretty much complete. It's already pretty much everywhere it's going to be by the end of that time. And that's really interesting when we consider this next map of genetic variation, which is looking at two mutations in different codons but in the same gene of the sodium channel gene. And these two mutations, which are in teal and orange hexagons, both provide very, very strong resistance to pyrethroid insecticides. And we only started using these in the 20th century, so sometime after the mosquitoes already invaded everywhere. So the question is, how exactly did these same mutations get all over the place? So there's two real main evolutionary mechanisms that can happen. You can have adaptive introgression. It can arise in one population and then spread everywhere else. Or you can have multiple substitutions of exactly the same mutation happening multiple times. And we can investigate this when we think of the sorts of patterns this would produce in a population genomic sense. So imagine this is a population, right? We've got chromosomes. We have one population that's got a linear genetic structure, and the asterisk shows where there's this SNP that gives a massive advantage in resisting insecticides. So what happens initially is that SNP will spread, increase in frequency, and even though that's one SNP, there's also this flanking region of maybe millions or even tens of millions of base pairs around it that will then get carried along with it. That's called genetic hitchhiking. The pattern it produces is a selective sweep. And you'll see that even though at the other parts of the chromosome, these individuals, these chromosomes, they retain their differences around that sweep locus, they're very, very, very similar. So lower nucleotide diversity and also low to GMAS D. So what happens? Some of these mosquitoes go into a new population, and then that same thing happens. And again, even no matter how different the rest of this new population might be genomically, around that locus, everything's going to look pretty similar. So this was what happens if it spreads via introgression. If you have multiple substitutions, it's like this. It starts off exactly the same, But when you have a second population, that same identical SNP is now on a different genetic background, and then that spreads. So within each of these populations, there'll be much lower nucleotide diversity, Tajima D, various other patterns that you can see. But if you compare these two populations against each other, they will look very, very different at that locus. So that's what I set out to do in this paper. This has just been published on Thursday, so have a look at it if you're interested. We've got those two mutations, teal and yellow. And so starting with this teal mutation, this is a latent factor mix model. Mosquitoes have got the correct number of chromosomes, which is 3. And you can see there's 2 peaks here. There's one on chromosome 2, one on chromosome 3. This pattern was basically the same when we built the model using the other mutation. I'm going to start talking about chromosome 3. This is the site of the sodium channel gene. These are where the mutations are. So of course we're going to see, what we're seeing are SNPs that are segregating in line with their resistance genotype rather than their population genetic structure at the rest of the genome. So of course we're going to see loads and loads of SNPs around that gene that are in line, that are segregating in line with the variation there. So there's these 2 mutations, but if you do a PCA, you actually get 3 genetic clusters of homozygotes. So there's 1 for the teal background, and then there's 2 for this orange background, and they split off quite clearly from each other. And then if you look at the individuals that make up those 2 clusters, and looking at them with FST, so on chromosome 3 here, you can see a big peak. This represents basically that there are 2 independent substitutions of exactly the same mutation on different genetic backgrounds that have then spread. So if we look within each of these backgrounds, well, this is Tajima's D among the individuals that have that have got that background versus wild-type individuals. There's a dip in Tajima's D exactly at that locus. We also see this more clearly in nucleotide diversity, and that's found across all of these sweeps. So what we're seeing here are the effects of this is basically a large effect locus. We've got a very, very strong selective advantage to having this. This is well characterized. There's lots of research has been done on these mutations in this gene. And so we're seeing this pattern as what we expect to see. And then the actual coverage geographically is pretty impressive here. You can see, for instance, that red background is in North and South America. It's also in New Caledonia. It's also in Malaysia. And it's also in Saudi Arabia. So it's literally spread all around the world. Now, we already knew quite a lot about the sodium channel gene before this. This is a very, very well-studied gene. This next bit was really quite surprising because there's another peak here. This is on chromosome 2. Why are there SNPs on chromosome 2 that are segregating in line with variation on chromosome 3? That was really surprising. Well, at this specific gene region are a cluster of glutathione S-transferase genes called GSTs. And there are 15 clusters along the genome, but it's this one cluster called the epsilon class where all of this interesting activity is going on. So these are well characterized in terms of insecticide resistance generally, but not in this exact way. So we knew that they were associated, but not necessarily that they had an enormous effect. And I'll show you what I mean by that. Well, so these basically there are outlier SNPs here. So if you're looking at the SNPs that are highly associated, looking just directly at those genotypes, and that's what you see in these rows here, there are individuals found across multiple populations that have identical homozygous genotypes at every single one of those outlier SNPs. And I've colored those in blue and green and purple there. And if you look at the distributions of those on a map, they've also spread all over some pretty large geography as well. So there's one that's across the entire Australian range, there's one that's all throughout Southeast Asia into Papua New Guinea, and there's one that's in the Americas all the way through the Pacific Islands also into Papua New Guinea. So these backgrounds, these are identical genotypes at all of these outlier SNPs. And if you compare them against each other as before, this is FST, there's an enormous peak exactly at that gene region, indicating that these are multiple different backgrounds. It's not the same thing. It's basically there's at least 3 different evolutionary events that have produced these backgrounds that have basically swept across this geography. And then if we look at, again, the Tajima's D at the top, nucleotide diversity at the bottom. This is a very, very similar pattern to what we saw with the sodium channel gene before. So again, sodium channel gene, very well characterized. We know it has a huge phenotypic advantage for the mosquito. In this case, these patterns seem to be pointing to a similar kind of thing going on with these sets of genes here. So basically, these results point to there being these two large effect loci for resistance, at least, well, there's probably more than two, but just two identified in this study, the sodium channel gene and then this GST gene as well, or set of genes. What's also quite interesting about these is that they're partial sweeps. They haven't fixed anywhere. Particularly for the GSTs, we didn't find a single population where these were actually fixed in the population. There were always wild types there as well. And what that says is that sometimes the wild type has an advantage over the resistant background. So there are either some times or some spaces where having resistance is costly. And that will be when insecticides are not being used. There will be a physiological cost to resistance. Again, this is well characterized in the sodium channel gene mutations, but we don't know anything about it for the other GST genes. And in a kind of fairly, almost ironic sense, there is another form of these partial sweeps in that some places like Saudi Arabia and Papua New Guinea, there are no wild-type mosquitoes left, but multiple sweeps have sort of got into the country at the same time, and then none of those are fixed because they're all basically, they have no advantage over the other backgrounds that have swept into there. So they won't actually reach, for each one of them, they won't reach a fixation because there are two or even three different backgrounds that confer basically a very, very similar advantage to the other ones that are already there. So this last bit is on what I was interested in, finding the sodium channel gene, chromosome 3, GSTs, chromosome 2, these both seem to have a big effect. We can look at that in a population genomic sense. It stands out. It really leaps out from the analysis. But there are also loads of other genes along the genome that we know will have some sort of different effects, maybe specific to certain insecticides, or maybe a general effect to multiple insecticides. And they will be interacting with these genes as well. So I explored that using linkage networks. This, again, we got 1, 2, 3 chromosomes, and the y-axis, these are histograms, basically, that are showing where do we see clusters of SNPs that are in extremely strong linkage with the sweep region, which is in that red bar there. So there are a couple of, where I've annotated these peaks that contain genes that we know from other studies are associated with insecticide resistance. And so this is one of the sweep backgrounds. If I show all 6 of them, what you can see is that the peaks are often in different places, and they're often for different genes. So what you can have are these 2 major gene clusters where there's been multiple evolutionary events happening, and then they've swept, but where they go, when they're locally in different populations, or associated with different sweep backgrounds, they will be locally interacting with different sets of genes to produce these mosaic phenotypes of resistance. And these, whatever might be, say, in linkage here, may be changing over time. This is really just a kind of cross-sectional snapshot of when we sampled. So I'll thank all of my co-authors and funders, and I'll leave you with this. This paper's just come out.
Any questions? I have perhaps one question. Have you reconstructed the time point of when the alleles evolved? And is it like, you seem to suggest that it's quite recent, but have they perhaps already evolved a longer time ago?
So in one regard, we don't expect that they would have evolved before insecticides would have been produced because they seem to confer a very, very strong fitness cost. Though some of the other, at least for these 2 major loci, but there may be other parts of the other genes along the genome that maybe were, we don't really know about the level of resistance, say, 200 years ago or something like that. But yeah, because our sampling occurred over about 10 years, we're not able to actually, I could estimate it, but I wouldn't necessarily want to be too sure about this being the case. For another species that we've also studied, which is the tiger mosquito, that one, it's a little bit clearer because resistance was recorded first in Singapore, and we can see its frequency is the highest in Singapore, and then it's spread in a very, very low frequency among a bunch of other locations nearby.
Thanks.
What's the cost to resistance gene when there's no insecticide?
Well, so for the GSTs, we don't really know. But for the sodium channel, basically the sodium channel works, pyrethroid, that's the target site for pyrethroid insecticides. So that will effectively hit the sodium channel, which produces an action potential for the nervous system. This kind of kicks open all of the sodium channels to make every single nerve in the mosquito fire at once. So it paralyzes it and the mosquito dies. When they get a mutation there, one of these mutations, that stops, it's like jamming the door, so the door can't be kicked open completely. So the mosquito's nervous system doesn't work as well, but it doesn't die from paralysis and whatever else when it gets sprayed with insecticides. So it's a very clear kind of, it's very clear why it's such a problem, but it's the only thing they can have sometimes to survive insecticides.
Okay, thank you.
Okay, so our next speaker before lunch is Alan Bergland from University of Virginia, and he's going to talk about water fleas and the effects of shared polymorphisms.
All right, thank you very much for the introduction. Thanks for the invitation to speak. So today I'll be telling you about our work studying balancing selection on shared polymorphisms between Daphnia species. Before I begin, I'd like to acknowledge that this work is the product of a PhD student in my lab who recently graduated, as well as various collaborators at UVA and elsewhere. Okay, so shared polymorphisms are polymorphisms that are found identical by state in multiple species. And there's different mechanisms that can generate or maintain shared polymorphisms. The theme of this session is convergence, so obviously convergent evolution can generate shared polymorphisms in multiple species independently. Adaptive integration, as we just heard about, can also generate shared polymorphisms between species. And additionally, shared polymorphisms can arise prior to the split of two species and can either be present because of neutral demographic processes, referred to as incomplete lineage sorting, or they can be actively maintained in both species via balancing selection. And these are oftentimes called trans-specific polymorphisms. Today I'm going to talk about shared polymorphisms in the Daphnia pulex species group. So Daphnia pulex was a species that was first described in the middle of the 19th century in Europe. It is an assemblage of many small-bodied species that live in ponds and lakes throughout the world. This is somewhat of a problematic species group. There was initially one species described. There's been at different times upwards of 10 or more different species described. Oftentimes they're given the same name in different parts of the world. And we'll be talking a little bit about that later today, or in this talk. But I want to just put a plug that Daphnia pulex is a really good species for studying shared polymorphism because of this interesting pattern of speciation that they observe, as well as the excellent genomic resources in this species group. So in this talk, I will be exploring the nuclear phylogeny of the Daphnia pulex species group, focusing on a handful of the taxa in that group. I'll then be focusing on two of those species and asking how abundant shared polymorphisms are, what are the mechanisms that can best explain the presence of these shared polymorphisms, and then the functional and fitness effect of one really intriguing shared polymorphism that we discovered. The data set that we used is whole genome resequencing data from over 2,000 clonal lineages from 6 taxa within this Daphnia pulex species group. That's Daphnia obtusa, Daphnia pulex, and Daphnia pulicaria, sampled in North America as well as Europe. Even though this data set contains these 6 different taxa, about 95% of our data come from genomes from European or North American Daphnia pulex. And for the remainder of the talk, we'll be focusing on polymorphic markers that were identified in or near single copy orthologs, and it's about 350,000 markers that we used. So one of the first things that we did was to build a phylogeny based off of these nuclear markers. One of the things that you can see right away is that these species, which have been identified as obtusa or pulex or pulicaria, are not reciprocally monophyletic. So, for example, and importantly, the Daphnia pulex species that are collected in Europe are not the same species as the Daphnia pulex that are collected in North America. This has been seen from mitochondrial analysis in the past, but this is the first demonstration of it using nuclear markers. European Daphnia pulex and North American Daphnia pulex are about 10 million years diverged. So it's a fairly deep and well-supported split. So for the remainder of the talk, we'll be focusing on European and North American Daphnia pulex. And when we examine the abundance of shared polymorphisms between these two taxa, we observe that there's tens of thousands of shared polymorphisms. And so what we want to do is address these four different mechanisms: introgression, incomplete lineage sorting, convergent evolution, and ancient balancing selection. So first, I will just tell you that shared polymorphisms between these two species do not seem to be explained very likely by introgression. This is the analysis of a ABA-BABA or a D statistic. And the estimate of the mixing proportion between these two taxa is very low. And so in comparison to some other contrasts we can make in this group, it is sort of the least well supported for evidence of introgression. So we can rule out at least recent introgression as a cause of those shared polymorphisms. Next, we wanted to explore whether these shared polymorphisms can be explained by neutral processes of incomplete lineage sorting. And to do that, we constructed two-dimensional site frequency spectrum. In the top left, you can see our observed 2D site frequency spectrum, and that gray or brown triangle in the center represents those shared polymorphisms, and about 1.8% of the mutations in our data set are those shared polymorphisms. We used moments to model a neutral demographic process and to estimate our expected 2D site frequency spectrum under a model of a population split, either just a split and no migration or a split with migration. And under either of those models, we see fewer shared polymorphisms than we would expect or than we observe under the empirical data. And in particular, there seems to be a deficit of shared polymorphisms that are common in both species. And you can see that when you look at the residuals. So in this case, the pink residual values indicate an excess of observed shared polymorphisms relative to the modeled expectation. So there seem to be more common shared polymorphisms between these two taxa than we would expect under neutral demographic processes, and therefore we can reject incomplete lineage sorting as a factor that is contributing to explain all of these shared polymorphisms. Okay, so next we wanted to address whether convergent evolution or ancient balancing selection, trans-specific polymorphisms, could explain these shared polymorphisms. And I just want to walk you through the tests that we did. So to assess these alternative mechanisms, we calculated allele trees surrounding these shared polymorphisms. And under a model of convergent evolution, we would expect that the distance between allele trees within a species between alleles would be short relative to the distance between species of the same allele. Right? So you can see the yellow highlight versus the orange highlight on the figure on the left. For ancient balanced polymorphisms, these trans-specific polymorphisms, we would expect that alleles that are within a species but differ by state would have a deeper coalescence time than alleles that are the same by state but in different taxa. Right? And so that is indicative of an ancient balanced polymorphism. And we can calculate these differences or these distances using the cophonetic distance, and then we take the difference of those two estimates within taxa and between taxa to calculate our test statistic. Okay, so panel B here is showing this difference of the cophonetic distance for our control, not shared polymorphisms, as well as our shared polymorphisms. And what you can see is that the vast majority of these mutations have allele trees which resemble the species trees. That's a negative cophonetic distance. And that would be indicative that these shared polymorphisms are present because of convergent evolution. However, there's one gene and a bunch of SNPs surrounding that one gene that show a positive difference, which is indicative of an ancient balanced polymorphism. These mutations surround a blue wavelength opsin gene, which is really intriguing, and we're going to dig into that right now. All right, so Daphnia are cool. They have a big cyclops eye in the middle of their head. They don't see very well, but they are able to use their limited vision to assess sort of light cycles as well as looming stimulus of predators. So the capacity to see likely affects fitness. We first wanted to explore the just patterns of genetic diversity and haplotype structure surrounding this locus. So in panel A, we're showing a sort of a schematic of the gene structure. The purple lines are different exons in that gene, and that pink box is sort of the region we're zooming in on, surrounding several exons where these trans-specific mutations occur. Panel B shows a linkage map of these mutations in that pink box. And what you can observe is that there is very strong linkage between many of these trans-specific mutations. Many of these mutations are non-synonymous, suggesting that they are functional. And then when you build that allele tree surrounding that blue light opsin, what you can see is that indeed the alleles cluster by allele type and not by species, again consistent with this idea that these mutations are very old. Okay, so this is an interesting gene, and we wanted to test the function of these mutations. And so we went back to our stocks. We identified European Daphnia pulex that harbor different alleles at this, or different genotypes at this locus. And because of the role of vision in movement in Daphnia, we wanted to first test whether different genotypes affect behavior and movement. And to do this, we set up an assay where we use trikinetics monitors. These are typically used for Drosophila work, but we use them for Daphnia. We put a Daphnia in the little tube, filled it with media, and these trikinetics monitors monitor a beam break, an infrared beam, as the Daphnia swims back and forth. And we put these trikinetic monitors into different chambers that either had blue light, white light, or were dark. We measured activity over a 12-hour window. And what we observed was that there were significant differences in activity between the genotypes. And what was really remarkable was that genetic variation was environment dependent. And importantly, there were shifts in dominance across different environments. So the heterozygotes were the most active in the white light treatment and the least active in the blue light treatment. And shifts in dominance have been implicated in the long-term maintenance of balanced polymorphisms, consistent with this idea of sort of long-term selection on this locus. Next, what we wanted to do is ask whether genetic variation at this blue light opsin affects fitness in the wild. And so we went back to a previous study we had conducted where we sampled a set of populations in southern England over multiple years. There's a lot of stuff in that paper I won't get into. But one of the really cool things we did in that paper was that we were able to reconstruct a multi-year pedigree of this population using whole genome resequencing. And in one of the years, in 2017, one of the ponds that we studied, D8, became dominated by two clones that we call A and C. And the size of these triangles or these diamonds reflects the abundance of these different clones. The A clone doesn't produce males, but it produces sexual eggs, so it's effectively female. The C clone produces a ton of males. And the pond went dry between 2017 and 2018. And so in 2018, all the individuals in that pond were F1 hybrids between A and C clones, right? It turns out that both lineage A and C were heterozygotes for this blue light opsin locus. And so in 2018, we were able to go and look at the segregation patterns at this locus and ask whether there's evidence of genetic overdominance at this blue light opsin. And what's really cool is we found it. So in these wild sampled individuals in that F1 offspring, we found that there was an excess of heterozygotes, right? So we would expect from a cross between two heterozygotes, 25, 50, 25. And that's what we see on average across the genome. At this blue light opsin, there is a strong excess of heterozygotes and a deficit of homozygotes, as you can see in panel E. Panel F shows the distribution of FIS, which is a way of capturing that deviation from Hardy-Weinberg. And that blue light opsin shows the strongest deviation from Hardy-Weinberg that we observe in the genome. Now, to show that this was not some sort of artifact of our genome annotation, we went back to look at lab-generated F1s, and we can show that in the lab, that blue light opsin segregates exactly as you'd expect from a double heterozygote cross. So this is direct evidence that there is a fitness effect of this locus in the wild, and that heterozygotes have a higher fitness in the wild than either homozygote. All right, so just to conclude, Daphnia pulex species group contains these cryptic species that are morphologically similar, called the same thing, but are not the same species. North American and European species of all of these different taxa are distinct and are in many cases millions of years diverged. There's a bunch of shared polymorphisms, but there's only unambiguous evidence of a few trans-Pacific sites, and that one of those sites is this blue light opsin that has fitness effects in the wild. And with that, I will thank my funding sources and take any questions.
Thanks for a really cool talk. I'm curious, have you modeled the blue light opsin with, for example, AlphaFold to figure out where these mutations actually are?
That's a great question. We haven't yet. The other thing that, if anyone knows how to do this, I've heard that you can actually predict the wavelength specificity of opsins by their protein structure. So I'm super into talking about that some more.
I'm curious about how recombination plays into that system. Do you see more shared polymorphism in low recombination region, or?
Yeah, that's a good question. We haven't characterized that. We don't have good recombination maps of this system. And because of the kind of the wacky clonal structure and the very small population sizes that these populations go through, they're effectively like kind of like QTL mapping populations in some ways. So it's something that we need to explore. It's a great question. Yeah. All right. Thank you all.
Okay, thanks.